and welcome to our online participants. Great to have you logging in for our live webcasting. It's my pleasure to introduce our speaker today, Dr. Vincenzo De Luca. Vince has been with us at Brock University since 2001, and he came to join us as a Tier 1 Canada Research Chair. And most recently, he's actually had that Tier 1 Canada Research Chair renewed with his area of specialty in plant biotechnology. Prior to joining us here at Brock in the Department of Biological Sciences, he was a principal senior scientist at Novartis Syngenta in the biotechnology world. And prior to that, he was a plant biology professor in Quebec at the University of Montreal. He's had so many achievements throughout his career. I'm going to really do some editing here, but I'd like to draw your attention to a few key points. Vince is currently a member of the Science Advisory Board at Vineland Research and Innovation Center, and he's also on the International Scientific Advisory Board at the Max Planck Institute for Chemical Ecology. He's been the past president of the Phytochemical Society of North America. He's authored over 90 scientific research publications. And although he's always been affiliated with us at the Cool Climate Oenology and Viticulture Institute, last year he officially became a team member as one of our COVI fellows. So with that, today Vince is going to share with us some of his research. And the title of his talk is Sustainability in Grape Production and Commerce.
Thanks, Dan. Well, thank you for the invitation to talk a little bit about the research that's going on in our laboratory. Today I'm going to talk to you a little bit about research that's funded by OMAFRA, NSERC, and some funding that's also obtained from a company, and therefore I've added industry to the list of supporters. The work I'm going to talk to you about today is work that's going on in my lab with Brent Weens, who's a student doing his master's degree. Work that Dr. Kyung-hee Kim, who's a postdoctoral fellow in our lab, and work that Dawn Hall has done. And she completed her PhD, and she's now working in British Columbia. The focus of the topic today, I'll start off with an introduction of what my research interests are, which really focuses on how cells become biochemically specialized for doing what they do. Plants have absolutely remarkable capabilities for this type of biosynthesis, where certain cells start to augment expression of certain pathways, and they become major factories for synthesis of a whole range of natural products. I'm going to focus on a couple of topics, a class of protein called acyltransferases, which are involved in grape volatile production. This really focuses on the well-known acyl esters of phenolic compounds and various terpenes. I'd also like to, while discussing this, I'll talk about a topic that's famous and infamous to some extent, the production of methylanthranilate, which is something that nobody wants in wine grapes, but which is produced in profusion in Concord grape. And hopefully our reasoning for working on methylanthranilate production will become obvious as I discuss the relationship between the production of anthocyanin 5-glucosides, which are actually diglucosides, and which are basically markers that people use to identify wines that are contaminated with grapes coming from North America or North American origin grapes. I'll also focus on another class of protein called glucosyltransferase and their role in grape natural product production. They play important roles in increasing the water solubility, the stability, transport, and compartmentation of these natural products, and of course play a really important role in allowing the accumulation of these and our ability to have grapes that contain them that are then used in winemaking. And I'll give you a specific example relating to a bifunctional glucosyltransferase that has really interesting roles in the biosynthesis of resveratrol, as well as the possibility that they're involved in the tartaric acid esters of phenols, which are important constituents of wine. And finally, I'm going to talk a little bit about some industrial work that we're doing that focuses on grape pomace and the secondary metabolites of grape pomace and their use as food additives and nutritional use. So I'd like to start my seminar relating to the issue of sustainability of grapes. We know that wine grapes are clonally propagated, and that as a result of their clonal propagation, what we are actually doing is growing large numbers of identical individuals that are growing together within a particular space. This, of course, leads to important issues such as increased susceptibility to a range of diseases. And it would be like we were cloning individuals, and we obviously are more susceptible to certain diseases than if we are a heterogeneous population. And this is really a phenomenon that's quite unique to wine grapes. And it's based on the fact that we don't have large-scale breeding efforts to try to improve grapes and create disease resistance, for example, simply because we need to clonally propagate these species. And there's a whole range of issues which you're probably well familiar with, why this is the case. And this has led to a heavy dependence on management techniques and the use of chemical pesticides to deal with disease issues. Over the last few years, there's been a tremendous increase in issues relating to regulation and the use of pesticides. Governments, particularly in Europe, we've seen an increased demand by these governments to decrease the use of the pesticides, decrease the pesticide footprint for wine grape production. This is basically because there are important contamination issues with respect to the land, the water, and the air, and the possibility that there are risks of these chemicals to the populations that live around these areas. And so, as a result, governments are asking for biological control mechanisms that can be used and can then lead to decreased pesticide use in order to grow the crop. And this, of course, leads to the need for genetically superior disease-resistant genotypes. And in order to do that, we have to breed crops. And one of the things that we're worried about is whether there are emerging diseases that are about to appear in the vineyard. One particular example is the presence of Pierce disease, which is increasingly prevalent in places such as California. And this is a disease that was not very prevalent in the past, but more and more we're starting to see this particular disease appearing in places such as that particular state. And this is known as an Eastern disease, and now it's found in various places in California. Same thing with powdery mildew. Are there new forms of powdery mildew that might appear that will be more vicious in the way they attack the grape plant? What about if there were new forms of phylloxera that had suddenly appeared in nature? When you read reviews on phylloxera, researchers are saying that there isn't a lot of funding for phylloxera research. We know that we generate phylloxera resistance based on the fact that we can create root systems that are resistant to the system. But if there was a new form of phylloxera, it would take some time to breed that resistance. So the types of typical breeding programs that exist in crops such as corn, wheat, barley, rice, etc., where we're always ahead of the diseases in terms of fighting resistance, this is something that is an open question with grapes because of the issues that I've just described. So if we're going to move towards breeding programs, what do we have to do in order to do that? Well, what has happened over the past 7 or 8 years or so is the use of genomics. There's been a lot of money spent to sequence the grape genome. And as you may know, Pinot Noir was selected for various reasons. And what's neat about sequencing of whole genomes is that you can use that genome to look at all grape cultivars because of the phenomenon of synteny. That means that the genes involved in various pathways, etc., seem to be organized in similar places on the 19 or so chromosomes, the 19 or so pairs of chromosomes that grape has. And therefore, the knowledge from Pinot Noir can then be extended not only to all the Vitis vinifera cultivars, but all the Vitis species that you find around the world, around the planet. And this then makes it very useful for what we call marker-assisted breeding. And I believe you've listened to a few talks in the last few weeks that describes that particular approach. The other tools that are available are remarkably sophisticated tools for doing metabolic profiles. In other words, looking at the chemistry of plants as the plant grows and identifying the periods when the grape, for example, will be manufacturing particular metabolites that are relevant for winemaking. We also have tremendous amount of work that's been done on pathways. And pathways are genes, pathways are proteins, and they are pathways that are important for winemaking. But pathways are also mutations. In other words, specific genes where there's been a change in the gene and the protein is no longer active. And the things I'd like to discuss today that are really important if we're going to be successful in creating disease resistance is that we have to be able to know the pathways that are important for some of the relevant products in winemaking. So for example, it's really important to know about the genes involved and the proteins involved in anthocyanin biosynthesis. And these are really very well known in a broad range of plants. It's one of the first secondary metabolism pathways to be characterized in many other species. And as you'll see in the next few slides, we know a lot about anthocyanin biosynthesis in grapes. Nutritionally important products like resveratrol. These are very well known in grapes, and I'll talk a little bit about that. The acylated aroma and flavor compounds that will give you the variation in organoleptic properties that the wine is going to produce are also important. And I'll speak a little bit about methylanthrinolate in my talk. Then there's C13 norisoprenoids, things like beta-damascenone and ionone, for example, which have all these floral characteristics. What I'm going to show you today is that we know a lot about how these things are made in grapes. But before proceeding with the grape story, I want to tell you a little bit about my area of research. You can see lots of models of pathways over here. And my major interest, as I said, is cell-specific specialization for secondary metabolism. We do a lot of our work in the rosy periwinkle, the Madagascar periwinkle. And if you visit our campus, you'll see large swaths in front of the greenhouse of Madagascar periwinkle growing. They're very beautiful flowers, but they're also very toxic plants because they produce these dimeric indole alkaloids, which are used in cancer chemotherapy. And our lab is heavily focused on how the plant is able to convert simple precursors into these complex structures at the end. Okay? But in addition to that, over the last 7 or 8 years, I've become increasingly interested in the grape system to look at the mechanisms by which the grapes assemble these interesting secondary metabolites over here. Why is cell specialization important? Well, this particular picture is a beautiful illustration of cellular specialization. We see these remarkable morphologies. This is an aromatic sage, which is used in cooking. And you'll notice that the leaf, this is a scanning electron micrograph over here. And this cross-section of the leaf shows you the leaf epidermis. And you notice that sticking out of the leaf, all these remarkable structures, this forest of hairs, which is produced as a result of cellular differentiation of the epidermis. And you can imagine an insect or other organism trying to get to the juicy part of the plant here. It wouldn't be very easy because of this forest of hairs. And not only that, underlying this forest of hairs is this big bag over here, which is called the gland. And this gland is full of aromatic terpenes. And you can imagine that these terpenes are released periodically from the gland. And probably they are not very interesting to the herbivores that are going to consume the plant. We like to use aromatic sage in our cooking, but we wouldn't want to eat large amounts of it as a raw material. So how on earth does this bag become full of the metabolites that are made? This is a picture of a mint gland. And what we can see over here is this remarkable factory for biosynthesis. As you can notice that it's found on the surface of the leaf. It contains a basal cell, a stalk cell, and you notice that with the darkening around there, that stalk cell is heavily suberized. And the reason why it's suberized in that manner is because it prevents any flow of metabolites in this direction into the plant. The flow of metabolites that are made is in this direction into the gland. So what happens in the mint gland leaf surface is photosynthesis takes place, carbon dioxide is captured in the presence of light, converted to sugars, and these sugars are mobilized all the way into these cell types, these specialized cells, these specialized factories called secretory cells. And the synthesis, the genes involved for menthol biosynthesis and highly methylated flavone biosynthesis are activated only in those cells and not in the surrounding cells. This is what we call cellular specialization. And as a result, it only takes 25 hours to fill this gland with these monoterpenes. Okay? And learning how cells become specialized for manufacturing has remarkable implications for our ability to use plants in all kinds of ways. Okay? So when we look at anthocyanin biosynthesis in grapevine, we know that most of the times the biosynthesis of anthocyanins seem to take place in the skin of the grape, and it seems to only take place at certain stages of development of the skin, and that means during veraison. And here you can see how complicated the pathway for anthocyanin biosynthesis is, but it uses precursors that are then assembled to produce what we call a central intermediate. In this case, it's a chalcone. And this is elaborated via a number of enzyme reactions. And you can see that as we make one precursor into another, it can go in different directions or it can continue to go down the biosynthetic pathway. And this leads to various types of flavonoids over here. Or if it's elaborated even further, it leads to the production of these types of anthocyanins. And we very well know that green grapes have really been derived from red grapes as a result of a mutation that took place sometime in history, where that mutation knocked out the ability of this pathway to make flavonoids, to make anthocyanins, and therefore gave us the green grape cultivar. We know exactly which genes are involved in converting a red grape into a green grape, and therefore we know how to convert a green grape back into a red one. In addition to that, we also know that depending on how we play with the decorations on these molecules, we will end up with a grape that is more red, which has the so-called cyanidins over here, or a grape that's more blue, which contains these delphinidins. You notice there's 3 hydroxyl groups that may or may not be methylated on this ring system over here. So depending on the ratios of these metabolites, you're going to get a grape that has a whole range of different colorations, from pink to orange to red to blue. Okay? And this is very relevant when we look at work that the Italians have done in the last couple of years, where they actually took a look at a whole range of cultivars of grape over here. As you can recognize, a number of Italian names, a couple of German grapes over here. And you notice that what happens from pre-veraison to veraison, some of the grapes turn progressively more red, or red to progressively more blue, and this Sauvignonace, which is a Tokai, I believe, remains green, simply because the regulatory gene that activates anthocyanin biosynthesis is mutated in that system, and therefore gives you the green grape. And you'll notice that when we go from red to blue over here, the ratio of the B ring, remember those 3 hydroxyl groups I was telling you about? You'll notice that the ratio changes from the red to the blue over here. Not only that, you'll also notice that this grignolino here has a lot of anthocyanins, but it has the cyanidin-based anthocyanins and not the delphinidin-based anthocyanins. So when you look at this result, you know that grignolino doesn't have a lot of hydroxylation ability to make the 3rd hydroxyl group, whereas these grapes over here have large abilities to hydroxylate and give you the 3, 4, 5 prime hydroxylation pattern. Okay? So this is what genetics does for you. It demystifies the genetics that are involved in creating the various genotypes that you see over here. And if we look at it based on this pigmentation pattern, we only see a couple of genes that appear to be involved. It's not a tremendous number of genes to differentiate at least the pigmentation profiles that we see over here. Okay? So the pattern of grape pigmentation in these 9 cultivars is regulated by differential gene expression of anthocyanin biosynthesis and their transcriptional factors. Now, if we go back down here and look at the difference between the red and the blue cultivars, we know that it's this hydroxylation pattern. But one of the other things that people like to define wines that are contaminated with North American genetics, let's say, is the fact that you see 3, 5 glucosides. Okay, if you start to see 3, 5 glucosides in the wine, you suspect that you've got American grapes in there. And winemakers don't want American backgrounds in their wine, because they want to keep their wines Vitis vinifera pure. Now, this is interesting, but if we could produce 5 glucosylated grapes, that would produce a more stable anthocyanin. And there are important commercial consequences to more stable anthocyanins in the winemaking process. Now, a few years ago, actually last year, these people actually identified the gene that's responsible for the glucosylation at the 5' position. Okay? And those of you who knew Dawn Hall, one of the projects she had was to go after the 5-GT. The objectives of the lab was to look at the 5-glucosylation and the methylanthrinolate step, because we know from the work that people have done that we suspected that the 2 traits are tightly linked to one another. Okay? So what these people did was to, the inability of most European grapevines to produce 3,5-diglucosides has long been used to classify wines according to their varietal origin. What they found was that the 5-GT that they found inside vinifera had 2 mutations that made the protein inactive. Okay? And when these mutations were corrected, of course, in bacteria, you, by magic, reactivated the 5-GT gene. And it's interesting that because there's 2 mutations, it's very difficult to find revertants in Vitis vinifera. Okay, if there had been a single mutation, I guarantee you, you would have seen a lot of revertants that you would have appeared, and it would have reappeared the 5-glucosylation. Okay. So what does this have to do with methylanthanylate now? This is what it has to do with methylanthanylate. When they looked at the Pinot Noir genome, they were able to show that the 5-glucosyltransferase was on the same chromosome as a gene that looked like anthranilate acyltransferase for making methylanthanylate. And it's only a short distance away. So you can imagine now when you're making crosses, it's very difficult to dissociate these 2 traits because they will be inherited together. Okay? So it tells us a lot about that vinifera has the capability of glucosylating this position, but it's mutated twice. And it's also telling us that if we could find the mechanisms to dissociate these 2, we could then dissociate the putative foxy flavor delivered by AMAT and getting the 5-glucosylation, so you could produce more stable anthocyanins in Vitis vinifera wines. Okay? Let me turn my attention to fluoronoisoprenoids, and this is work, you've got the publications over here if you're interested in that. If we look at how norisoprenoids are made, and these are the class of floral compounds such as these that we see over here, and we notice that they are actually derived from carotenoids. Okay? And this particular protein called Vitis vinifera carotenoid cleavage dioxygenase actually cleaves the bonds of these various carotenoids, and it releases these C13 compounds that are the backbone for the assembly of these norisoprenoids. Okay? What is fascinating, of course, is if we take carotenoids and we consume them ourselves, we would end up having genes or proteins that will cleave this right down the middle. And this would be really important to produce things like the vitamins that are important for our eyes. Okay? So when we look at where carotenoids are assembled in the plant, you'll notice this is another beautiful scanning electron micrograph, I must add. And here's a typical cell in the mesophyll, and when you focus on this part of the cell, this is a chloroplast. And carotenoids are all made inside chloroplasts, and they're really important in chloroplasts because they help chloroplasts to do photosynthesis, but they're also a source of the carotenoid, and these proteins must be associated with the membranes of the chloroplast, and when they're made, they become associated with that, they harvest the carotenoid and convert it to this derivative over here. So what they found was that Muscat grapes make more isoprenoids than Shiraz. I'm sorry about the write-up over there. But they also found that there's only one gene in grape that's responsible for this reaction. So you can imagine if you want to manipulate the levels of these metabolites, you would want to be able to use this as a marker in your breeding program. And that would allow you to end up with varieties that have higher levels of this particular reaction, and then you would hope that you would end up with higher levels of this end product over here. However, there is a caveat. If you can't supply this, you can't really make lots of that. So one of the issues is, are there sufficient carotenoids inside developing grapes so that the supply of substrate is available for making this particular intermediate. So there's two things that you really have to screen for when in your breeding program when you're looking for higher levels of these metabolites. Okay? So here's another example of a remarkable genetic tool that can be used for creating new grape cultivars that have altered levels of these floral volatiles. Now let's go to the methylanthranilate story for a moment. Some of you have seen this slide before, and you can see that the manufacturing of methylanthranilate is not terribly simple. It requires the supply of a substrate that is normally involved in the assembly of one of the aromatic amino acids called tryptophan. And during grape development, the levels of this particular metabolite has to rise. And it seems to rise during veraison. And this HPLC profile shows us that there is an anthranilic acid pool. And this is very unusual because anthranilate normally doesn't accumulate in plants in any great level. It's usually completely converted to tryptophan. But in developing ripening Concord grapes, we see anthranilic acid pools rise. And it's got to be converted into this activated intermediate called anthranilicyl coenzyme A. In addition to that, it needs to be supplied with methanol, which is going to be the methyl group donor in this reaction catalyzed by the protein called AMAT. And you'll notice that in order to produce methanol, you may know that all fruits have methanol in them, and they usually have their methanol when they're ripening. The reason why methanol levels rise when they ripen is because they're starting to break down their cell walls. And cell walls are composed of a magic molecule called pectin. And when you produce proteins that will hydrolyze the methyl groups that you find on the pectin, with pectin methylesterases, you get the release of methanol. And so there's no way that you can make methylanthranilate unless you have both methanol and the CoA ester that you have over here. One of the other things that really fascinated us when we looked at this was the fact that the vast majority of the protein that we were able to find was in the fruit itself, but near the skin, rather than closer to the inside. And we know that during veraison, when you're walking through a Concord grape field, you have these methylanthranilate aromas that waft in the atmosphere. And many of you know, possibly, that methylanthranilate is very unappreciated by birds. And so birds don't like to go into methylanthranilate-laden Concord grape fields. And hence, maybe that's one of the reasons why this trait evolved in Concord grape, although it has to be proven. So we went back and we said, well, the European grapes have the methylanthranilate producing capability, or at least the gene that looks like the methylanthranilate producing capability. And this is work that Brent Wiens has been doing during his master's. And we notice here that he looked at a whole range of grape cultivars. And I know that we have Grand Noir over here that doesn't look like it's got it. We will go back hopefully this fall and repeat those experiments and see if it is present in Grand Noir. And we'll have more information about that. But what it tells us is that virtually all the cultivars that we look at seem to produce a product that might be an AMAT like protein. Well, we decided to focus on 4 cultivars, the Chardonnay Musqué, Cabernet Franc, Shiraz, and Sauvignon Blanc, to clone the relevant genes. And here you are. I mean, this was probably several months' work. I'm sure Brent doesn't appreciate how few slides I'm dedicating to him. But you can see here that you have a product that's about the same size. And this gene product can then be cloned and popped into a system that allows you to produce protein. And strangely enough, when we went back and produced the protein, we got this result over here. And what we noticed is that the Chardonnay Musqué, the Cabernet Franc, and the Shiraz all produce a protein product which is quite small. In contrast, Sauvignon Blanc produce a protein that's about the right size. And this protein is about 95% identical at the amino acid level to Concord AMAT. So remarkable conservation of these genes in terms of the way they look from between North American and European species of grapes. So we went back and said, well, is the protein found in the plant, in the grapes? And so we went back. These are using antibodies to this protein, and you notice that Sauvignon Blanc produces a protein product of the right size, whereas you also see these smaller products over here. But Cabernet Franc doesn't seem to produce, and Chardonnay and Shiraz don't seem to have that product. When we looked at the sequence, however, you'll notice that there's a base pair missing over here. And interestingly, all three of these have this mutation, and it introduces a stop codon, and therefore generates the truncated protein that we see over here. So Sauvignon Blanc makes a protein of the appropriate size and possibly a truncated protein as well. And the other cultivars only accumulate the truncated protein. So one question we have is, do these have biological activities? And what's the biochemical role of Sauvignon Blanc, AMAT like protein? So when you look at this story, we notice that there's one fully functionally active form of acyltransferase in grapes, in European grapes, and there's one mutated form that produces a truncated protein. So when we're looking at commercial cultivars of grapes, we should go back there and scan all commercial grape varieties for the presence of the active form during grape ripening, which is something we're going to do this September, I guess. This will allow us to document the genes that are expressed versus the useful wine metabolites that are produced, and will produce a fingerprint of the genetic makeup of each of the cultivars that we have. Okay? And interestingly, it can then also be used as a marker for introduction of the trait in other vinifera cultivars, either by breeding or by the verboten word genetic engineering. Let's turn our attention to grape glucosyltransferases. These are really remarkable proteins because there are hundreds of them in grape. They have important roles because they increase the solubility and stability of secondary metabolites such as anthocyanins. These glycosides may be transported or stored within vacuoles, and they may also be activated forms of a molecule which can then act in further reactions and in biosynthesis. So if we look at resveratrol over here, I thought I might as well give you a short lesson in biosynthesis. And you'll notice that resveratrol comes from common precursors, the aromatic amino acid known as phenylalanine, and it's converted to phenolics over here. And you know that these phenolics are responsible for the formation of caffeic acid, ferulic acid, phenols in general. But if you involve the conversion of this to this intermediate and the resveratrol synthase, you end up in the formation of resveratrol. So it's derived from phenylalanine, is a common pathway to flavonoids and anthocyanins, and it the formation of this is done by a unique polyketide synthase known as resveratrol synthase. But after resveratrol is produced, it always tends to be glycosylated. And you can see the types of glycosides. You can produce trans-PCID or trans-resveratroloside over here. So when we looked at the glucosyltransferase involved, not only did we find it, but we found out that it had a bifunctional role. It not only was it able to glucosylate the carboxyl groups of all these phenols over here, it also was able to glycosylate resveratrol as well as a number of other flavonoids. And what's interesting is it was more active at one pH towards resveratrol and at a different pH for the glucosylation of these carboxyl groups. So it's able to make two different types of glucose-containing natural products. So what we've done with the glucosyltransferase, and this is in collaboration with colleagues in France over here who are very interested in this, was to use the resveratrol glucosyltransferase as a tool to manufacture this glucoside. And very little is known about how valuable or useful this type of glucoside might be in terms of its use in various foods, its absorbability and stability in the human gut. And so one of the things we would like to see is further work to see if this could be of any commercial relevance. And that, of course, was cloning of the gene from the plant and popping it into microorganisms for manufacturing this type of thing. On the other hand, because it has this bifunctional role, is it possible that it might also be involved in making these caffeol ferroyl tartrates, which accumulate at significant levels, over 30 milligrams per liter in the wine? And we know that they have important roles in wine preservation. They also have negative roles in terms of some of the oxidation properties of these. And if we look at caffeol tartrate over here, it looks a lot like malic acid, doesn't it? The difference is the presence of an extra hydroxyl group in the tartrate. So one of the things we'd like to do is to find out if indeed the synthesis of these tartrates is similar to the synthesis of cinepoyl malate in Arabidopsis thaliana. These genes have been discovered in Arabidopsis, and we know that these glucoses are activated phenols that can be used in this reaction to produce cinepoyl malate. And in Arabidopsis, huge amounts of this compound accumulate within the leaves of Arabidopsis. Therefore, there's no reason to think that this might not also be possible, because we have documented the presence of these glucose esters in grapes, and therefore the conversion would involve the identification of this gene, which would be a similar one to this, in producing these tartrates over here. Now, identifying that gene would give us a marker for the tartaric acid esters that would be produced in grape. How could that be used in improvement? You could either use it to increase or decrease the tartrates of interest in your product. In the final part of my talk, I'd like to tell you a little bit about the work that Kyung-hee Kim is doing in the lab, and the nutraceutical value of grape pomace. So one of the things that we tried to do was to identify the antioxidant activity and other health benefits, the various polyphenols that you find inside this powder. And we also looked at free and bound forms of these things. One of the things that permitted the rapid analysis of this was this new system that we have in the lab called ultra-performance liquid chromatography. And you'll notice that HPLC traditionally will run 32 to 48 samples in 24 hours because of the long run times. This system has been standardized to run in 7 minutes, and that means you can run about 200 samples in 24 hours. It makes a big difference in your ability for high throughput or medium throughput screening. If you're interested in particular peaks, you could also use it in a much shorter time frame, and you can see how many runs you could do. So the very short runtime suggests that the system can be used for a whole range. I mean, breeding, mutant screens, and the program can be automated to select promising candidates in your research. So this is some of the analyses that Kim did. And you can see here all these metabolites that you find in pomace powder versus wet pomace or in seed pomace. And what's important to note here, this is a 7 to 8 minute run. Okay? And these are the various phenols that were identified in each of those pomaces. Same thing was done with phenolics. And you can see them separating nicely in each of the pomaces that we're looking at. And we did a comparative analysis of each of these. And when we look at the concentrations of these major phenolics, you can see that they're found in wet powder, wet pomace, and seed. And they're also found in the residue after you extract. So this gives you a sense of how well you've extracted the material. And you can see that these things were hydrolyzed afterwards in order to get the phenols. So they're treated with acid and so on and so forth. And you can see that there's various molecules that are found in the pomaces over here, and they're quantitated. When we look at these pomaces, we also see that there's significant amounts of various anthocyanins. You can see the peaks coming out over here, and they're found in both wet pomace and in dry pomace. But you can see that upon drying, the levels of this triply substituted dimethylated malvidin derivative has gone down. Okay? And this information, of course, is going to be used by the industry in relation to how they're going to, the information that they're going to put on their packages, for example. One of the other metabolites that we looked at was triterpenes. And you can see here that this is just standard oleanolic acid over here. And we can see that we find lots of this in dry pomace, in wet pomace. We also find it in the seed over here. And we also find it in grape skin. And usually this oleanolic acid will accumulate on the surface of the grape. And it has important antiseptic properties and helps to protect the grape against pathogens and so on, and bacteria, for example. It's interesting that the seed coat has very little of this particular metabolite compared to inside the seed. So what did we find in the pomace metabolites? We also found certain grape pomaces that have significant amounts of resveratrol. Okay? And so some pomaces have nothing, and others have significant amounts. In addition to that, they also have significant amounts of this molecule known as viniferin. And the French like this molecule because they want to sell it to people for skin cream production and so on. So not all pomaces are created equal. If we summarize now, you can see that we find major amounts of phenols. We find significant amounts of procyanidins, delphinidins, various anthocyanins, as well as large amounts of oleanolic acid. And when you read the literature, the Gallo wineries have patented processes for extracting oleanolic acid from grape pomace, of all things. So they're trying to get value from the pomace and using oleanolic acid for various commercial processes. So let me summarize. What I've just talked to you about today is the remarkable advances that we've made in our knowledge in the biosynthesis of things like anthocyanins, resveratrol, AMAT like aroma compounds, C13 norisoprenoids, phenolics, and terpenes. So that information is there. It needs to be applied now. And the issues whether to breed or not to breed need to be resolved. And I know those issues are much more difficult to solve than the breeding program that we're proposing. These tools are going to be used to fingerprint differences between cultivated wine grapes. This is very useful in marketing and commercial protection of cultivars. Very useful and important for identity preservation. So far, it seems that the documented differences that appear between different cultivars are probably relatively small. They're going to only involve a few genes. So I don't know how mysterious Cabernet Sauvignon and Pinot Noir are going to be once we find out that the differences are not as great as we had anticipated. They're going to be used in breeding programs, whether used in classical genetics or genetic engineering, I'm not sure, but they'll be used to improve color, flavor, and aroma. And they're going to be used to improve stress tolerance in grapes. Now, if you're going to improve cold, drought, disease, or herbivore tolerance, you want to be able to have these traits following the other traits. So that's why these markers are very important in the breeding program. The grape pomace analysis and its potential uses as value-added sources of antioxidants. Well, we hope it'll allow improved marketing of byproducts for incorporation into foods. Hopefully we can create new health claims with this. And again, we can use it for identity preservation and protection of certain grape pomace sources. In finishing, I'd like to thank Kyung-hee Kim, Brent Weens, who are in my lab now, and of course Dawn Hall, who was an important component of my lab for about 5 years in her life. Thanks very much.
Thank you, Vincenzo. We do have time for some questions from the audience. Maybe I'll start us off.
The Grand Noir, is that the Grand Noir that we provided you with?
The Grand Noir is the Grand Noir that you provided us with. That's right. So we'd like to go back this fall and harvest some more and maybe do a larger sampling over different stages of development and take a look and see if it's there at all. And as we do a broader range of screenings, we'll be able to find out varieties where it's just not expressed, or cultivars where it's just not expressed.
We suspect that it's a hybrid, and yet it hasn't got any diglucoside either.
Yeah, well, I mean, if it's a hybrid, it should have the diglucoside.
Well, no, there's been few hybrids which didn't have diglucoside.
Yeah.
Like some of the French hybrids.
So,
few,
yeah, so it may mean that you've bred out the active protein. It's something that can be verified now that we know what we know about the particular gene in question. I expect it could be doable to find out why certain hybrids don't have the ability to make diglucoside and others do. And I always thought there were trace amounts of diglucoside in some of these grape cultivars.
Yeah, but even in vinifera, I understand there's some traces.
Right.
Because we had done with Guelph some work, and they were sort of worrying about this that they could find, and I've seen in the literature as well.
Yeah. And
the methods of analysis now to detect what they thought didn't exist.
Right. And it's probably based on the fact that acyltransferases often have, broad substrate specificities, and there may be another enzyme in vinifera that allows that reaction to take place at a very low rate, and therefore you're able to see, if we incubate our proteins long enough with substrates, they will add more than one sugar. So the system can produce trace amounts of diglucoside. The big question is I firmly believe that if you were able to incorporate the diglucoside producing capabilities without the other components or traits that you don't want, it may improve the stability of your anthocyanins. And that may have important consequences for the color production in wines. So that's why I'm saying if you could separate the Foxy from the the 5GT, it might be an interesting direction to take the breeding program.
So the Grand Noir here would be further away from Concord than Chardonnay. Is that what?
Well, not necessarily. What it's telling us is that the Grand Noir may not have that particular gene. And what we have to go back and look is what exactly were the crosses that were made and the back crosses that were made to generate Grand Noir. And that's easier said than done, I believe, from what you're describing, because it's suspected, but nobody knows exactly how it was developed, right? But you know, this is just the beginning. We've got to go back and look at these in detail now. Well, I'm sure you've seen this with the work that you've done with Judy. It's given us insight, but now we have to get to the bottom of it, and by doing the experiments we hope to do this fall. The fact that the methylanthranilate, or the gene that makes these esters, is so closely associated, that means all viniferas have these associated to one another on chromosome 9. And so now we have to go back. And the other thing that this tells you is that there's a lot of mutations in grape. And that mutation makes that particular protein non-functional. And we need to find out more about that, because what is it, when you generate the mutation, was it due to hundreds of years of clonal propagation, for example? Was that part of the reason why you generate the mutation? We all know that as we age, we accumulate mutations, which is part of the reason why we don't live beyond a certain age, right? And grape, we've got grapes that have been around for 500 years, 600 years, and they're clonally propagated. So what mutations did they, and we've actually selected new cultivars based on mutations that have appeared in the field. So a lot of that is that generic to the original species, or is it something that occurred with human intervention and cultivation? Those are, I think, some of the questions that I think the people in France and Italy and Australia and so on, who are going to try to use the genome to address those issues. Where do our grapevines come from, and how did they become what they became?
I have a question. For the Sauvignon Blanc, where you found the AMAT Like gene that produced that protein,
Right.
do you have any insights yet, enzymatically, what volatiles it may be targeting, or is it too premature yet until you start to
Yeah.
profiling of the metabolites during?
Yeah. One of our problems is accessibility to gas chromatography, as you know. We have lots of GCs in Brock, but they're all heavily used. And so the question now becomes, how are we going to do the profiling and relate it to the gene expression? We're working hard to try to produce a functional protein in bacteria, and then we can identify the types of substrates that it will use. And but without knowing what the profile of Sauvignon is, Sauvignon Blanc is, we need to match the two so that we can go back and say, okay, this protein is made at week 14 to 18, and look at this, it seems to correlate with the appearance of this volatile, just like we did with methylanthranilate. I mean, in that case it matched perfectly with the appearance. And I would guarantee you that if we produced the methylanthranilate, let's say we took the Concord grape methylanthranilate gene and stuck it into vinifera, by genetic engineering, you're going to make brand new volatiles that you never saw before. Of course, you would have no other genes that came from Concord then, right? It would be a single gene that would go in, and you may end up with really interesting aroma profiles that then you have to ask yourself, how do you generate that without genetic engineering? because genetic engineering is not yet accepted.
Are there any other questions? Well, with that, I'd like to thank you, Vincenzo, for an exhilarating talk this afternoon, and I'd like to present you with a token of our appreciation.
Oh, thanks.